Hi everyone. This video is for the Cell Cycle Lab. So as always, pull up your procedure and make sure you read it all the way through. This is a pretty straightforward lab today. The big thing you're going to need is either a bag of M&Ms, a bag of Skittles, or maybe some coins that you have around the house. You'll need about 100 of these. So the Cell Cycle Lab is going to start with a review, as per usual. And we're looking at the central dogma. So if you forget a little bit of this, just take a look at your central dogma chapter in the molecular cycle of study. It wants to know, in your own words, what is the central dogma of biology? Make sure not to copy verbatim what the internet or the textbook says, but put it into your own words, okay? It also asks for a few different terms. So number 2 says, when DNA codes for mRNA, or messenger RNA, what is it called? Where does it happen? And then the second step of that, when the RNA codes for an amino acid, what is it called? And where does that happen? So where in the cell are those two things taking place, and what do we call them? And number 4, it says, what is one of the primary functions of the cell? Well, the cell has a lot of functions, but this is asking for the primary function. So in this case, think about the most important thing that a cell can do. What does it make? To give you guys a hint. What's the most important thing that a cell will do? It's going to make something. So go ahead and write that down. And then part B is going to start talking about the cycle of life, the cycle of the cell. Okay, so this is going to be revisiting the Cell Cycle Lab, but it's also going to be touching on a few extra things that we haven't learned about just yet. Now you should recognize this picture. This is a nice visual of the cycle of a cell's life. Of course, we know interphase, it's about 90 to 95% of a cell's life. During interphase, it's doing the job of a cell, whatever type of cell it is. It's going through gap 1, synthesis, and gap 2. Maybe it's pushed off into the gap 0 phase, just to work, not really to worry about growing or dividing anytime soon. And then if the cell is going to divide, it will enter our cell division phase, which consists of mitosis and cytokinesis. Of course, mitosis, remember we have 4 parts to that: PMAT, prophase, metaphase, anaphase, and telophase. And so make sure you remember, as you think about this lab, all the different components and what happens during each cycle. So this is a nice little reminder. Again, we like to use the labs as a review, a really good way to study, okay? And then you have a lot of text here, so make sure you read through all this. It says the cell cycle is critical. It helps control against genetic damage. It helps prevent against uncontrolled cell division, right? We have checkpoints as we go to make sure that things are big enough before they divide, that DNA was copied properly. The cell cycle is regulated with cyclins and cyclin-dependent kinases, or CDKs. So these are types of proteins, right? ASE means an enzyme. And so these guys are the ones controlling the checkpoints, making sure that everything is in working order before letting it go to the next stage. When the cyclins and CDKs are not working properly, if they're not doing their job of checking, the cell can divide more often than it should, causing this exponential type of growth, which could result in tumors. So tumor cells typically just keep dividing, they keep dividing. They have extra blood supplies going towards them to help feed the cells, and they can get pretty large pretty quickly. So having those CDKs and cyclins, those proteins that are helping to regulate the cell cycle, is very important. Now it says often when cells are too large and the cell membrane cannot adequately meet the needs of a cell, the cell will divide into 2 daughter cells. So this goes back to our surface area to volume ratio that we talked about. If you guys remember that egg and the egg cube that you put in your food coloring water. Remember, as the volume gets big, the surface area goes down. So very big cells don't have enough surface area or membrane to move nutrients in and out, which means that they could starve or they could die of waste buildup. So as a cell gets too big, it's going to signal to itself that it needs to go ahead and split apart so we can get more surface area on a smaller cell, right? In multicellular organisms, this is called cell division. It consists of mitosis and cytokinesis, right? Of course, as we grow, going from an embryo to a baby to an adolescent to a full-size adult, our cells are dividing so that we're getting more and more cells as we get larger. If the organism only has one cell, if it's a unicellular organism, maybe it's a bacteria, an archaea, a protist, then the process is called something different. It's called binary fission. So we've seen this term before. Binary fission is much faster than cell division in a eukaryotic cell or a multicellular organism, simply because there's less DNA, there's less organelles, there's less to it. So binary fission is a rather fast way to reproduce, and especially with prokaryotic cells, they don't have long strands of DNA. If you remember, instead they have a nucleoid. They have a circular strand of DNA. It's much smaller than what we have in our cells. And so it's very easy just to copy the circle of DNA so that they can split and have two different organisms, right? For us, when our cells divide, we just get more cells in our organism. But of course, if a unicellular bacteria divides, it's then two separate bacteria organisms, okay? So we're looking at an example of E. coli here. E. coli is, of course, a bacteria. You've probably heard about it a lot because it can make people sick if we're not careful about how we handle our food. And so E. coli goes through this binary fission process. Now remember, it's very fast. So some bacteria cells can replicate very, very quickly. And we're going to look at an example of this. So it says E. coli is a bacterium, okay, right? It's a prokaryote. It can double its population very quickly under the ideal conditions. So of course, it's still a living thing. It still needs nutrients, a way to expel waste. It needs space and energy and all of that. If it's given all of that, it will divide pretty fast. So we're starting with one E. coli cell, just a single organism. And let's say we place it in a petri dish with some nice agar, some nutrients, everything it needs to survive. That single E. coli cell is going to replicate. It's going to copy its DNA through binary fission. It's going to divide and form two different cells. Those two cells are then going to take in the nutrients, they're going to copy their DNA, and they're going to produce another cell each. So now we're up to 4 cells, right? I now have 4 cells. Each of those 4 cells can divide itself to get 8 cells, right? So as you can see, it can double its population very quickly. Now in our example, our time frame is every 1 hour. So every hour, our existing E. coli population will double the number of cells because they're undergoing binary fission. So take a look at this table. You should see a pattern, right? Every 1 hour, you see the population doubling because they're all dividing. So complete the table. Of course, you have 7 boxes, so you're going to go up to 7 hours. How many do you have throughout that whole time? So put down your number of E. coli. It was started for you. You should be able to finish it pretty easily. And then you're just going to graph that. So you want to graph your data from that table. Put time on the x-axis, the horizontal axis, and try to spread it out. So you're going from 0 to 7 hours. On your x-axis, try to use up as much of that graph paper as you can. So instead of putting an hour with every single box, maybe use up 3 or 4 boxes for every hour, okay? That will help you get a better visual on your graph. Your y-axis then is going to be your population number, your number of E. coli, okay? And of course, we started out with 1. Make sure you go all the way up to what you ended with so you can fit them all on the graph. You should just have one nice line. You're going to plot all your dots. You can connect it together as a line. And it says this could represent bacteria growing inside a person's gut. It could also be very similar to how cells would grow in a tumorous state. So when they're reproducing very quickly. Just a nice visual to see how some of this works in a very short time frame, right? 7 hours is not long at all. All right, once you finish with that, we're going to do a little bit of experimentation today. So part C, we're going to start talking about exponential decay, okay? So in part B, we talked about exponential growth, things growing very, very quickly, all right? Now we're going to switch gears a little bit, and we're going to talk about things decaying rather quickly, all right? So if we have these cells that are growing way too fast, our CDKs, our cyclins, are not working to stop them during our checkpoints. We as medical practitioners want to have some way to combat that, to help people with these different tumors and cells that are growing out of control, right? So one way they do this is actually inserting a radioactive pellet right next to the tumor cells. They do this because, of course, most of you know, if you pump chemotherapy into the whole body, it's a toxic chemical, right? Its point is to kill cancer cells, but it can also have some pretty drastic effects on the whole organism, okay? It's pretty hard to go through. And so if we can put that radioactive pellet right next to the tumor cells, we can lessen the effects on the whole body. It can be more centralized. So we're looking at this, of course, from a therapy perspective, trying to think about how we can maybe kill some of these tumors using radioactive pellets. But first, we need to think about what radioactive decay even is. So it says treating cancer can be very difficult. Anything from surgery to radiation therapy to chemical therapy, or a combination of those can be used. One form of radiotherapy is called brachytherapy, or internal radiotherapy, if you've ever heard of that before. And so this is that radioactive pellet. They place it very close to the tumor, and they allow it to undergo a radioactive decay right next to the tumor cells. These radioisotopes have many uses in our modern society. So in this example, they're helping to kill the tumor cells nearby, but we use this type of thing in a lot of different ways. For this lab, you're going to need your 100 M&Ms, Skittles, coins, really anything that has two sides, one of which is marked differently than the other, okay? You'll also need some type of container to put them in and to shake them. You want a flat surface, so your procedure suggests 2 plates that you're going to stack and shake. You could also use a large container if that's easier. Just make sure you have a lid, okay? All right. Now, starting off, we want to think a little bit about this radioactive decay and what it actually does. Do you guys remember isotopes? If you remember isotopes, hopefully you do, an isotope is a variation of an element. It has the same number of protons, same number of electrons, but a different number of neutrons, right? Neutrons are in the nucleus. They account for some of the mass of the cell, but not the charge. They're just heavy. So we have certain types of isotopes that are naturally occurring for the different elements. Carbon has been one that we've talked about a lot. Carbon-12 is the most common naturally occurring isotope. Of course, you know that just by looking at the periodic table, because the atomic mass of carbon is around 12.011 AMU, which means that most of the carbon we find will have an atomic mass of 12, okay? Of course, this means there's 12 protons and 12 neutrons because the atomic mass is equal to the protons plus the neutrons, okay? But 12.011 is higher than 12, which means that every now and then we find some carbon atoms with more mass to it. Carbon-13 and carbon-14. They're going to exist at very small amounts. Carbon-14 especially, you don't find it very often, but it does exist. Carbon-14 will have 2 extra neutrons, right? So it says carbon-14 is a radioactive isotope of carbon. Radioactive, think it's not very stable, right? It's not going to hold its shape very well. It's going to start to break apart. It says it's considered radioactive because its nucleus is not stable and it will eventually decay. Depending on the element and the isotope, it will decay at different rates, right? The rate of the decay might change. It says radioactive decay is not a chemical reaction. Remember, chemical reactions occur with the electrons. Those valence electrons that we kept talking about, that's a chemical reaction. But now we're actually talking about the nucleus of the atom breaking down. So this is a nuclear reaction. Carbon-14 is made naturally in the atmosphere when cosmic rays from outer space push a proton out with a neutron, slamming into the nucleus, and turning nitrogen-14 into carbon-14. How the heck could that happen? Well, remember, the number of protons is what determines what type of element it is. So if we take a proton out or we add a proton, we change the element, okay? So essentially what this is saying is we have a lot of energy coming in from outer space, right? The sun, of course, we always have rays of radiation coming into the Earth. So we have energy coming from outer space, and it's actually moving a proton out of this nitrogen-14. So nitrogen-14, remember, atomic mass is equal to protons plus neutrons. So we have 7 protons and 7 neutrons. If we move the protons around and we only have 6 protons, well, we've changed the element. Now it's carbon, okay? Draw this. So number 1, it says draw the nuclei of nitrogen-14 and of carbon-14, okay? However you want to do this, I would probably draw some circles all clumped together in your nucleus. Maybe you put some pluses on the protons, right? However you want to draw it, put the nucleus, you don't need to worry about the electrons, put the nucleus of nitrogen-14 and carbon-14. Okay, now that we've kind of given a visual for what's happening in the nuclear reaction, we're going to model this with our M&Ms or our coins. So number 2, this exponential decay is what we're going to be looking at using our model. Each M&M or penny is going to represent 1 radioactive atom. During the experiment, we're going to record our results on a table and then we'll graph it at the end. Okay, so every single M&M you have, or whatever your object is, that is 1 atom. And remember, these are not stable. So over time, they're going to start to break down. When they break down, they don't actually disappear. They just turn into a different element, okay? So for our purposes, this experiment is going to help us model what it looks like when these turn into different elements, when they are no longer carbon-14, okay? You're going to place your 100 M&Ms or pennies in a certain order. So maybe if you have a coin, you use heads as your marker. Whatever you decide, keep it consistent, okay? If you have Skittles, one side should be marked with an S. Whatever you have, you want to place all 100 as flat as you can on your plate or your container with your marked side or your head side facing up, all right? Make sure that you spread them out and they're not stacked on top of each other, especially if you're using coins. Now, if you have a container, just put the lid on top. If you have a plate, you're going to turn the plate upside down and put both faces together so that you make a nice contained unit, right? Now, how we're going to do this is you're going to take your container or your 2 plates together and you're going to shake them twice. That's it. 1, 2, okay? So shake them twice in a gentle up and down motion and then set them back on the table. Take off your lid. And you're going to look at all of the different candies or pennies and you're going to see which ones have flipped over. So the ones that have flipped over, you no longer see the head or you no longer see the marking on the candy. Those have decayed. The nucleus has changed and now it's no longer the same element. So you're going to take all of those that have flipped and you're going to take them out of the container and put them off to the side, okay? Anything that is flipped over, maybe it's now on a tails side or it's on a blank side of the candy, take it out. Make sure you start recording this into your table. So at trial 0, it's written down for you. At trial 0, we had 0 M. I think they might have put this backwards. So make sure that you take a look and maybe you swap these answers. So it says trial 0, the very start, our M marked side up, we had 100, right? It told us to put all of them face up. So that should be 100. So go ahead and change that. And then M or marked side down, we had 0 of those to start with, right? Okay. You're going to continue this until you have removed all of your coins or your M&Ms, right? Which means you're going to shake it twice, write down how many you have, remove the ones that have flipped over until you aren't left with anything. If you do need more space on your table, that's fine. Maybe you just enter a few more below, okay? Or write it off in the comment section if you need to. But get all the way through until you get to 0. All right, so once you've finished and you've removed all of your candies or coins, you're going to graph it. Now, depending on which side you had up to begin with, if you had all the M's up to begin with, or if you flipped them over and you had all of your tails or your blank sides up, that's going to change your table up above, of course. Okay, so either you leave it the way it is, or you flip it if you did it in the way that I had said. When you go to graph it, you only need to graph one side that represents the atoms that have not yet decayed. Okay, so when you when you shake them and they flipped over, those ones have decayed, so you're not worrying about graphing that portion. You want to graph the atoms that have not yet decayed. So you're starting out with 100, and it's slowly going to be decreasing. So on your graph, make sure you put time on the x-axis. Time will really be your trial for your table up above. Okay, we're going to think about it in terms of time, but this is your trial number. Graph that on your x-axis, put all of your dots, and then you can connect them with one line so that you get a nice visual trend on what this would look like. Now below the graph it says, all right, we did this in trials, right? We had trial 1, trial 2, trial 3, but this really represents time. And so for this experiment, we're saying that each trial represents 1 day. So thinking about this in terms of the time it takes for half of these to flip over or to decay, it says how many days did it take for about half of your sample to flip over? So if you started out with 100, how many trials did it take for you to get to 50 of them left? Okay. Was it 1 trial? If so, it was 1 day. If it was 2 trials, then it's 2 days, right? So how many days did it take? Go ahead and write down your answer. You can round to, if it took just a little between 1 and 2 trials, go ahead and put that down as well. It doesn't have to be exact calculation, that's okay. Somewhere between whatever trial it was. This is known as a half-life. Okay, a half-life is the amount of time that it takes for half of your sample to decay. So in this situation, every time you shook your container, about half of the atoms probably decayed, okay? So probably, if you shook it, your very first trial, about half of them probably flipped over and decayed, okay? During 1 day, or trial 1, what is the probability that a particular candy or penny will flip over? What do you think about that? You have 2 sides to it, right? A front and a back. So, what is your percent chance that it would have flipped over? Think about flipping a coin in the air. What's your chance that you get a heads? Write that down. Number 11, it says, is it possible to tell which particular candy or penny will flip over? So with certainty, when you start out with 100, can you point to any given one and say, this one's going to flip when I shake it? Yes or no? And number 12, is it possible to confidently estimate how many will flip over? So this is all about percent chance, right? Again, if you have a coin that you flip, what's the chance you're going to land on a heads? You probably know that, okay? So can you confidently estimate how many will flip over with each trial? I would think so. So think about that for a minute, okay? We can estimate, maybe not with certainty, which exact one, but we can estimate how they're going to flip in rough numbers and percentages. So down below we see a graph. It should look rather similar to what you did up above. This graph is the half-life of carbon-14. Okay, so carbon-14 as an isotope, this is about how long it takes for it to decay over time, right? Now, the half-life for carbon-14 is 5,730 years, right? That means if you started out with 100 carbon-14 atoms, after 5,730 years, only about half of them would still be carbon-14. The other half would have decayed and turned into something else. So every time you see that section of gray lighten on your graph, that was another half-life, right? Another half of them decayed. So after one half-life, we're down to about 50 of them remaining. After a second half-life, we're down to 25 of them remaining, right? 50 divided by 2. After another half-life, we're down to 25 divided by 2, or about 12.5% of them remaining, okay? That's how half-life works. So it says these do not disappear, right? They're just turning into another type of element. Matter and energy can neither be created nor destroyed. Instead, they just kind of change form, okay? So using this graph given to you for carbon-14, how old is a sample of carbon-14 if you only have 12.5% of it remaining? They use this a lot in archaeology to tell how old samples are. So when they find a human skeleton, they can look and see if a certain isotope is remaining, how much of it is still there, and they can estimate how old it is, how many years it's been around. So half-life is really great for things like that. So if we have a sample of carbon-14 and only 12.5% of it is remaining, how many half-lives have we gone through? And once you know how many half-lives we've gone through, how many years old is the sample? So this answer, make sure you put it in how many years old it is, right? And you're going to do that by thinking of how many half-lives there were and maybe multiplying by one half-life length. Or maybe you just look at the graph up above, right? Where is the 12.5? What is the year that it lands on? It's going to be a pretty old sample. And number 14, you want to complete the table, okay? So we have a 400 gram sample of carbon-14. 400 grams, right? It has the number of half-lives at the top of your table. So go ahead and fill that out. We start with 0 half-lives. We go to 1, 2, 3, 4. How many years does that correlate to? So at 0 half-lives, we've gone through 0 years, right? At 1 half-life, we have 5,730 years that have passed, right? That's given to us, okay? What about after 2 half-lives? How many years have passed? You can do the math, right? You can multiply 5,730 times 2, or you can look up above at the graph and use that. Do that for 3 and 4 as well. And then we're going to think about this in terms of fractions and percents. Okay, so at time 0, before anything is decayed, we have 100% of our sample, which of course 100 is 1 over 1 as a fraction, right? So of a fraction, we have 1 or a whole unit, 100% remaining. The amount that we were given to start with was 400 grams, so we still have 400 grams of it. Okay, so that's your first column completed. After 1 half-life, half of it has changed, right? We're only left with half of it, which means, what's your fraction? 1/2, okay? 50%. 1/2 equals 50%. So after 5,730 years, we're only left with 50% of the sample, which means we take 400 grams divided by 2, or times 1/2, equals 200 grams. So after that length of time, we only have 200 grams left. Complete the rest of the table. So after 2 half-lives, how many years have passed? What is the fraction of it that's left? To do this, you could do 1 half-life times another half-life. So 1/2 times 1/2 equals 1/4, okay? What is 1/4 as a percentage? And what's 1/4 of 400 grams? How much do you have left? Continue that for 3 and 4 half-lives. And number 15 says, how old is a substance that has 50 grams left of carbon-14 from an original 400 grams? So if you started out with 400 grams and you only have 50 grams left, how old is the substance? Well, you could look up at your table, right? Think about how much mass it took to go from 400 down to 50. How many half-lives did you have to go through? Well, 400 grams, divide it in half, you get to 200 grams, that's one half-life. 200 grams, divide it in half, you get to 100 grams, that's another half-life. Get to 50 grams, how many half-lives did it take? But this is asking how old it is in terms of years. So once you know how many half-lives it took, multiply that by 5,730 years to get your answer in years. How old is it? How many years old is it? And now we get to our analysis, all right? So your analysis, you should be pretty familiar with how these work now. We're going to think through some different problems. There are many different types of radioactive isotopes, all right? Some have very long half-lives. It takes a very long time for these things to decay, whereas others decay very quickly. For example, potassium-40 has a half-life of 1.828 billion years. So that means every 1.28 billion years, half of them have decayed. It's a very slow process. It helps us date really old objects, things that are much older than 1 billion years, right? Beryllium-14, another isotope, has a half-life of 4.84 seconds. That is an incredibly quick decay, right? Probably pretty dangerous. So number 1, it says if you have 40 grams of radioactive beryllium, how much is left after 4.84 seconds? So the half-life of beryllium, as we just read, is 4.84 seconds. So after one half-life, we have half of it left, right? You started out with 40 grams. How many grams are left after that first half-life? Well, 9.68 seconds is just another half-life, right? So how many grams are left then? And then what about for 19.36 seconds? So you're going to have to do a little bit of math here. You're going to have to figure out how many half-lives have passed after 19.36 seconds. Maybe you do this by taking 19.36 and you divide it by 4.84 seconds, okay? How many half-lives do you have passed? And that will tell you what percentage or fraction you have left of your sample. And then you just take your fraction or your percentage and you multiply it by your total 40 grams that you started with to see how much you have left, right? Probably not a whole lot is left at this point. You've gone through a number of half-lives, so it's been decaying for a little while. Very short while in this case. Number 2, we're going to look back at carbon-14. So the half-life, as we know, is 5,730 years. Let's say that a sample of carbon is taken from a specimen we find on an archaeological dig site. That sample has 1/16 remaining. 1/16, right? How old is it? How many years old is it? You might first want to find out how many half-lives have passed. So if we started with 1, remember 100%, a fraction of 1 over 1. One half-life, we were left with half. Another half-life, we were left with a quarter, all the way to 1/16. How many half-lives have passed? You can then take that number of half-lives and multiply it by 5,730 years to get the total number of years that have passed. So how old is it? How many years? And then number 3, you're going to graph a function, okay? It's not as complicated as it looks. F of x just means it's a function of x, meaning we could plug in different values for x. So in this case, we're graphing 1/2 to the x power. We're going to start out with x equaling 0, so 1/2 to the 0 power. Then we'll go to 1/2 to the 1 power, 1/2 to the 2nd power, 1/2 to the 3rd power, all the way 1/2 to the 5th power. Okay, so complete this table. Your first column is the number for the x value, going from 0 all the way up to 5. Okay, and then your second column there is what the function equals. So 1/2 to the x. When you plug in the x value, what is your answer? Hint, it will probably be fractions, right? So you can write it out as a decimal, or I would just write it out as a fraction because I think it's a lot easier to see the numbers in the pattern that way. You are then going to graph that table, right? So don't let that be overwhelming. Your x value, you can put on the x-axis. So start at 0, go up to 5, okay? The y-axis, you're just graphing the values that you got in that second column, okay? So x being 0, you start with 1/2 to the 0 power. What does that equal? Plot that on the y-axis. And then scoot over to where x equals 1. 1/2 to the 1 power, what does that equal? Go ahead and plot that on your graph, okay? Once you plot all of your points, connect them with a line so that you see a nice, good visual on what this looks like. And then you're going to do this again with a slightly different function. So your function now is f of x equals 2 to the x power. You're going to do it the exact same way. You're going to go from 0 to 5 for your x values. And then in the second column of your table, you're going to write down what that value equals. So x to the 0 power equals what? X to the 1 power equals what? All the way down to x to the 5th power equals what number? Then you're going to graph that. X values 0 to 5 on the x-axis, and y values from your second column for the y-axis. Connect your dots with a line once you finish. Now under that graph it says exponential functions have the input as the exponent, the x value. Exponential growth functions will have a base that is greater than 1. Exponential decay will have a base that is between 0 and 1. What does that mean? Well, for your first example, you got a lot of fractions, right? They're between 0 and 1. For your second example, they were all bigger than 1, weren't they? The base cannot be negative, 0, or 1 for the exponential functions. So these are some really nice visuals on how we can have exponential growth, meaning things are growing very quickly at an exponential rate, and how we can have exponential decay, how things are breaking apart quickly at an exponential rate. Now, I shouldn't say quickly necessarily, because as we saw, sometimes they can take billions of years to decay fully, right? But sometimes they can happen pretty quickly as well. So whatever the half-life is, however long it takes, that's what you're going to be seeing on your graph. This graph that you made, both of them are just generic ones for any type of half-life that we could assign. Now, we think about this, of course, with tumors, with trying to kill the cancer cells that are growing way too fast. They're growing exponentially, doubling in size very quickly. We can also think about this with bacteria cells, maybe, that are growing very quickly, exponentially, right? We also have a half-life in medicine. So every medication you take has a half-life, meaning that after a certain amount of time, your body has metabolized half of it or gone through half of it, okay? Some medications, say you only take a medicine every 2 weeks. Your half-life for that medicine is probably 2 weeks, right? But say you take something like ibuprofen and it says to take it every 4 to 6 hours. Well, your half-life for ibuprofen is probably between 4 and 6 hours, meaning after about 4 hours, you've probably metabolized through half of it. So then maybe it's time for another dose so that it's effective, okay? So half-life is how doctors think about how much to prescribe and how often to prescribe medication. How much can the body take based on your metabolism? right? Pharmaceutical companies test this out, and that data is then provided to the medical community so that they can properly give dosages to patients without overdosing them and causing harm, or underdosing them and not really having much effect at all. So half-life is an incredibly important concept for medicine, for pharmaceutical medicine especially, right? How long will it take for your body to use the medicine that you're given? When will you need your next dose? Okay. All right, you have a few more questions on your analysis. What are the two major components of the cell cycle? What are the three stages of interphase? Okay, you might have a fourth stage, but if it's off to the side, don't worry about putting it in. Number 7, what are the two stages in cell division? And then number 8, it says what are the 4 major stages of mitosis, right? So just a good review, make sure you write those down, use this to study from, okay? And once you finish with that, it's the end of your lab, right? So I hope you guys enjoyed it. As always, ask questions if you have any, okay? This is some pretty interesting concepts, I think, so make sure you go back and you review this lab before your competency exam. All right, have fun.